I'm very happy to introduce Professor Edward Marcotte, who is an evolutionary biochemist whose research broadly uses tools for proteomics, bioinformatics, and systems and synthetic biology to study the biochemical mechanisms of human diseases. He has authored more than 250 publications. He has patents. He has received awards from the National Institute of Health Directions Pioneer. He is an elected fellow of the American Academy of Microbiology, the American Association for the Advancement of Science, and the Royal Society of Chemistry. He has co-founded the single molecule protein sequencing company ELIXIR, and he's a professor in the Department of Molecular Biosciences at the University of Texas, where he holds the Mr. and Mrs. Corbin J. Robertson Sir Regents Chair in Molecular Biology. So, Edward, it's a pleasure to have you here, and with that, we give you now the spotlight so you can start.
Great. Okay. So, welcome to everybody who's joining online, and thank you to Gonzalo and Arne and Christine for inviting me to talk to everybody in the 3D Bio community. It's exciting that you're all getting together and sharing talks and science and building a community, and it's nice to be able to participate from afar. So, as Gonzalo said, I'm joining you from Austin, Texas, so it's still a bright morning here for those of you in Europe. I know it's getting into the afternoon there, and I'm going to tell you a little bit about our research on trying to map kind of all of the core eukaryotic protein complexes as a step towards getting towards 3D structures for all the kind of core machinery in eukaryotic cells. But I want to start by acknowledging all of the people that have contributed to the work that you're going to see, and a particular shout out to Rachel Cox and Ophelia Popoulos right here, who led the computational and the experimental work that you're going to see for the first part of the talk, aided by sort of a growing number of very able colleagues, both in my lab and in that of my long-term collaborator, John Wallingford. And in fact, I think I spied Anna Battenhouse is online joining us right now, and I think Ophelia maybe as well. The work was also a collaboration with some groups outside of the University of Texas, including at Boston Children's, Illinois, Arizona, and Carnegie Mellon. And I'll try to call out to people sort of and certainly label contributions as we go through the slides. And then at the end, I'm going to bring in some work from Katie McCafferty and Gabe Hugerbrugge and a collaboration with Hui Bui. Okay, so let's get started. So, assuming my slides advance. There we go. Okay, so I'll just sort of give you a little bit of a perspective for where we tend to sit in research, which is we're sort of at the intersection of this triple Venn diagram, where my group tends to work on a lot of questions that relate to human disease genetics, especially in a few areas more than others, such as ciliopathies or the kind of developmental disorders that arise from the failures of cilia. We explicitly take a very evolutionary perspective, and so we bring in data from many different organisms, often sometimes sort of crazy organisms, to try to get interesting vantages on this, or even just to study intrinsic questions of evolution itself. And as Gonzalo said, the kind of tools we use would be proteomics, bioinformatics, systems, synthetic biology, and we kind of ping pong back and forth between computational and experimental strategies very explicitly to try to make rapid progress kind of in both domains where possible. So you'll see a little bit about that today. And the topic that we're going to be talking about is, there we go. Okay, I have to figure out how to advance my own slides. Zoom changes everything. So we're going to be talking about sort of this central problem of in the human genome. Of course, we have about 20,000 odd protein coding genes, and a few of those we know quite a lot about. So I think the estimate is there's 2 papers a day published on the tumor suppressor p53. But for a very large fraction, we actually don't know very much detail about what those proteins are doing. And of course, we would like to be able to fill in a lot of the missing holes and all the missing biology and biochemistry and cell, biology and genetics and so on about what the roles of these proteins are in the human species. And this sort of gets worse as we move into thinking about all the mutations in the genes, where even for genes where we know a lot about what the encoded proteins do, we don't necessarily do a very good job predicting the effects of mutations. So two mutations in the same gene can give rise, for example, to completely different diseases in many cases. And understanding this and when it's consequential or not is a key central question. And the thinking is that better estimates of the roles of the proteins in the cells, how they interact, how they assemble into higher order complexes, and the three-dimensional structures of those assemblies is going to be a key step in being able to understand our own genetics and make much stronger predictions about the effects of all of the different mutations. So this is sort of our central interest in all of this. And all of this essentially comes down to this reductionist view of human disease genetics. So to a first approximation, all human genetic diseases can be linked to a molecular etiology. And I'm showing three examples here of multi-protein complexes for which this is the case. So is the SWI/SNF complex, what's called the BB-some, and the KRAS-RAF complex. And so, for example, if you mutate any of the genes encoding the proteins in the BB-some, you get this debilitating multi-organ syndrome known as Bardet-Biedl syndrome. And this plays a role in the cilia, and this is a ciliopathy. And similarly, if you mutate KRAS or RAF, you get a different syndrome, Noonan syndrome, and so on. So we understand a lot about the function and sort of causal relationship of these to the emergent diseases. Well, by far, all of how they give rise to all the multi-organ effects. But, you know, it's to the level that, for example, and this is one example out of 1,000 I could have chosen, we know that if you mutate a particular arginine 632 in the BBS2 protein, then it's been shown in the literature by Ludlum, Aoba, and coworkers that blocks the physical interaction with a second subunit of the complex, and it disrupts the BB-some complex, and that disruption disrupts its role in the cilia and leads to Bardet-Biedl syndrome. So we can sort of trace from the genetic defect all the way forward using the structure and interaction information as the key to decode the genotype to phenotype relationship. Now, there's something else that's special about these three complexes, and one of the reasons that these were chosen is that, in fact, these are evolutionarily ancient. And so each of these three complexes, the subunits, were present all the way back about 2 billion years ago in the last common ancestor of all eukaryotic cells. So these systems, besides being key to very important disorders in contemporary humans, were templated a couple billion years ago in the canonical kind of origin of eukaryotes, modern eukaryotes. So the biochemistry itself is ancient and the assemblies are ancient, but the context that it operates on has changed through the course of evolution through multicellularity and building the mammalian lineage and so on. And so that now in the contemporary world, mutations in these proteins, protein coding genes are linked to these disorders, but obviously LICA didn't have a Bardet-Biedl syndrome, but it would have had some LICA equivalent of the Bardet-Biedl syndrome when there was a failure to that. Okay, so we're very interested in this intersection between the structure function on human protein complexes and their evolutionary origins and being able to triangulate back to describe kind of the core conserved working machinery of eukaryotic cells, which is what we're going to be talking about today. And to put this into context, this is a little piece of the eukaryotic tree of life. So here's the plants on the right and animals on the left over here. So you're here at the red star, moving down into fungi, through opisthokonts into slime molds. So these are the amorphia. And then in the middle, we have single-celled eukaryotes, like the ones that would live in swimming around in pond water or would opportunistically try to kill you, like Plasmodium. What all of these have in common, excuse me, is of course, we all derive from LICA, this last eukaryotic common ancestor, on the order of about 2 billion years ago. And so that has all of these interesting consequences for the conservation of the machinery. In principle, we can study that core machinery in any of these organisms and learn it from looking at many different systems and for understanding the genetics. And I'll make the case with this beautiful analysis from Rachel, in which she tallied up the causal genes for different human genetic diseases. This is taken from the OMIM database. So each dot is a set of genes that's linked causally from good, reliable clinical genetics information to these diseases. And the x-axis is the number of genes in that set, and the y-axis is what fraction of those genes are in evolutionarily ancient families and date back to LICA. And what you'll notice is this huge pileup on the top left. So that box is 72 distinct diseases for which every causal gene known is traceable back to LICA. And this is anemia and Noonan syndrome and things like this. And so we sort of have this interesting situation, this dimension to human disease, where some systems linked to these diseases are young evolutionarily. So pituitary hormone's an animal-specific innovation. This is a disease of an animal-specific hormone. But some diseases are diseases of ancient systems, and in this case, an ancient organelle. Ciliary dyskinesia is a failure in the cilia, just like short rib thoracic dysplasia. And the components of this were laid down mostly back in LICA, and all of the genes that we link to that disease are also evolutionarily ancient. So we like to explore this relationship back and forth. I'll say a little bit about where we get this information, since this is for a computationally sophisticated audience. So this is an analysis getting these LICA gene sets from the group here you see in a nice collaboration with Maureen Stolzer and Danny Durand at Carnegie Mellon, in which we've taken the predefined gene families. In this case, we're using the orthologous gene families defined by the eggnog database. And we've applied phylogenetic models to those in combination with a species tree from the Quest for Orthologs Consortium to try to project back in time under different models. So modeling duplication and loss and/or horizontal gene transfers to make an estimate of which of these families were likely present in LICA. And depending on the details of the models, is essentially 6,000 to 10,000 of these orthogroups that date all the way back to LICA. And they span roughly 2/3 of the human genes. So out of the 20,000 human genes, a little over 13,000 of those are in these LICA orthogroups and about 5,000 orthogroups. So think 2 to 3 human genes per orthogroup is about the scale we're talking about. So there would have been some duplication, along the way to the current human lineage of those genes, but they would project back to this core set here. Okay. So what was LICA? Because we have this set of genes that we can assert were in LICA based on the phylogenetic evidence, we have a pretty good idea of what the overall composition was. And so this is a cartoon drawn. We're actually using this nice, if you've ever seen Swiss biopics in UniProt, there's this sort of showing your localization in cells for proteins. I love these iconized versions. And so we actually took the icons from Swiss biopics and assembled a LICA. And each of these cartoon objects links back to a set of genes that we find in LICA by the phylogenetic analysis. So it would have had the necessary elements, mostly necessary elements of being a eukaryotic cell, a nucleus, a mitochondria. It was a swimming organism, so it had one or more cilia. Good evidence in the literature kind of arguing that maybe there was more than one cilia. But what's really striking, and it had a remarkably sophisticated endomembrane system. So the endoplasmic reticulum, the Golgi, the vesicle trafficking, endocytosis, exocytosis, secretion machinery was all present essentially all the way back in LICA. So very, very sophisticated eukaryotic cell with all of this internal machinery that we retain a large fraction of in our cells today. Okay, so we wanted to be able to say more than just draw a cartoon and to really start saying something about what are these core eukaryotic machinery that are the sort of the common heritage of contemporary eukaryotic cells. So can we say what the core set of proteins and protein assemblies and moving towards protein structures is that's shared across eukaryotic cells, including us? And so we set out trying to experimentally map these by looking at modern eukaryotes and projecting back to find the conserved interactome. And so for this, we needed a technique that would work on any sample and any tissue. And that meant that we had to avoid recombinant DNA strategies and avoid antibodies. And instead, we turned to techniques that would be very general. And this meant for us sort of old school grind them, find them biochemistry, where you grind things up in liquid nitrogen and separate. But rather than an old school biochemistry where you're following an assay for a single protein or activity of interest, we do mass spec on every single fraction in a separation. So we're sort of gently separating out the protein complexes as best we can without denaturing them. Mass spec on every fraction, and we measure where all the proteins co-purify. And then the notion is, if you repeat this, separating on different principles, so different biophysical properties like size and charge and so on, then we can start building up very strong statistical evidence that proteins are bound to each other regardless of how we separate, or maybe which tissues we look at, or which organisms we look at. And so we can build up very strong evidence for stable protein complexes where we're learning this information for all proteins simultaneously in an organism. So it's a experimentally intensive, instrument intensive technique, but it's also very high throughput in the you're essentially multiplexing the information about all of the complexes simultaneously in those samples. So we've been developing this approach now over a few years. So we've been sort of stepping up in complexity. We had a kind of a first big screen. It was a very nice collaboration with Andrew Emily's lab, in which we were able to get an initial draft of human protein complexes from a couple thousand biochemical fractions on different cell lines, analyzed in this fashion. And then we were able to then extend that again with Andrew's lab to about 6,000 biochemical fractions analyzed this way across different animal tissues to start getting at conserved animal protein complexes. And then at the start of COVID, my lab dug in and really tried to extend this into the plant world, where we really had very sparse protein interaction data at scale for the whole proteome scale. And so Claire McWhite and Ophelia and others in the lab, Kevin and others, ended up analyzing a couple thousand biochemical fractions from lots of different plants to get conserved plant protein machinery. And I'm going to take a few examples from this to give you a sense for what this data looks like to try to build some intuition about it. Okay, so we're going to look at some data for two protein complexes, and we'll just sort of get a feeling for what kind of information this data brings you. So we'll look at the proteasome and the CCT chaperonin. So proteasome has 2 copies of each of 14 unique subunits, and CCT has 2 copies each of 8 unique subunits. And we'll just take a few of those subunits from each, and we'll sort of look at what happens when we take, in this case, a Arabidopsis sprouts, and we grind them up in liquid nitrogen and separate by size. So we're doing this in a way the complexes stay intact. And if we do a mass spec on every fraction, and we look at the traces here, I'm just showing 3 subunits of each, you can see that the proteasome subunits come off together because that's where the proteasome elutes. And the CCT subunits, boy, this is much noisier, but you can see them co-eluting here, and there's something going on over there as well. Okay. Now, if we take the same sample and we separate by a different property like charge, now we have different separation properties, different elution profiles. Again, subunits of the same complex co-elute, and they co-elute differently from subunits of a different complex. If we repeat in a different tissue, maybe there's different interaction partners or different splice isoforms. Again, we have coherent behavior within the bound proteins and differential between complexes. So this starts to be a signature of what it means to be a member of this complex versus that complex. And as we add more data, we get stronger and stronger signals. So think about something like GWAS. As you add more and more individuals to the statistical analysis, you can start to draw very strong inferences. And this really gets true as we add the comparative evolution dimension. So this was Arabidopsis, now 5 subunits each, but we could do the same thing with broccoli. So Ophelia went to the grocery store and ground up broccoli florets, which are a beautiful source of plant nuclei, and many other plants. And now we really start getting this sort of very, very strong signature. You can see the strong coelution of each of the subunits across evolution in a conserved fashion, differential. And from these data, we can then clearly define the components of these complexes. Now, the study we've been doing now is we've taken these historic data, the plants and the animals, and we've supplemented them with a couple thousand new biochemical fractions from filling in holes in the phylogenetic tree of eukaryotes and adding available data from other groups around the world where they had done comparable experiments, as in Plasmodium. And the data set now, we have about 10,000 individual biochemical fractions analyzed from 150-ish different biochemical separations across the species in this tree here. And this is, we actually pull down mass spec data from groups at Harvard and in Europe, which is primarily from human and mouse. And so all told, we're quantifying about 128 million unique peptides through the course of this big analysis. So because this is shotgun mass spec, we're analyzing tryptic peptides. And so we have information about isoforms, which versions of proteins are being expressed in the data, and frequently about modifications, as well as the abundances of the proteins, as well as their participation in different interactions through the course of the separation. So these are extremely rich data sets with information about the expression, interactions, and modifications of the proteins that we could spend years analyzing the richness of these data. And of course, all available. So if people are interested in this, all of the data are publicly available. So here's what the CFMS data looks like. You can think of this as a matrix of 10,000 fractions. And remember, we're doing this in a comparative framework. So we're no longer looking at individual proteins, but rather we're looking at protein families across species, so orthogroups. So we have 6,000 orthogroups that are all estimated to be in LICA by the phylogenetic analysis. And here's a blow up of subunits for 3 known complexes. So here's the proteasome again, and you can contrast that with this eukaryotic initiation factor 3. And I'm just pulled out a few of the various elution profiles for different organisms here. But again, you can see the kind of coherent separation behavior of subunits of the same complex and differential with another one. And so this is the signal that we would then mine to determine the interactions. And as a reminder, these have a precise correspondence to physically separable biochemical purifiable entities. If we wanted to, we could dig into this fraction here and maybe follow with a few other separations and possibly purify and characterize this structurally. For now, we're inferring the association of the complexes by analyzing the patterns in the data. So how does that work? So mass spec, we need a reference database. So we need a genome or a de novo transcriptome for every organism. So many of those we have, but for the cases we don't have a genome, we actually either sequence the genome or sequence a de novo transcriptome and find gene models. And then we have to assign those into the orthogroups. This is one of the reasons we like the eggnog orthology definitions, because it's very easy for us to take the eggnog models and apply them to any new species. And then once we have our proteins sorted into orthogroups, we can then interpret the mass spec data. We actually do that at the level of the orthogroups. So we have this ambiguity we have to deal with where, say, on one evolutionary lineage like humans, maybe a gene was duplicated and we have 2 separate protein coding genes encoding that functionality. But say something like wheat is hexaploid, we might have 6 separate protein coding genes. And through a lot of testing, we realized that if we actually combine the information at the level of the orthogroup, we get much better signal for determining this kind of evolutionary families and their interactions. And so this gets us down to orthogroup level elution profiles for all the protein families. And then we do machine learning to learn the interactions. And in practice, what this means is all of the known protein complexes are captured in our data internally in the exact same measurement as the new ones. So we have the known protein complexes to serve as internal test and training controls to train a supervised classifier to distinguish which pairs of proteins are part of the same complex versus not. And we have to be a little careful about data stratification and things like that, but it turns out to be a reasonably straightforward supervised classification task. And we can learn to identify protein-protein interactions from these data because it's a data-strong area that we're in, and we can control the false discovery rate extremely well. So this is the performance of a classifier on a withheld set of known protein interactions, measuring the precision and recall at recovering the pairwise interactions. And you can see we can control the false discovery rate extremely well. So this finally gets us to a set of interactions among conserved proteins. And we do the mining in a way that we actually require evidence across the evolutionary tree to varying extents. And so we have now an estimate of the core eukaryotic protein interactions measured from this data set that would correspond to the LICA interactome. It's really hard to draw a picture of something as complicated as this. So this is an attempt that we've been developing over a couple of papers now for how to represent these interactomes. The features we're trying to capture are that, of course, we have direct evidence for binding of different pairs of proteins, but you can tell from the data we're actually getting sets of proteins that are working together. And there's a natural hierarchy to binding relationships where two proteins bind and assemble a subcomplex that assembles with another subcomplex to make a larger one, and accessory factors come in. And so we're trying to capture that. So this is a hierarchical clustering of our interactions designed to capture that. Each colored dot is one of these conserved protein families, these LICA orthogroups. And where there's a circle around them, that's evidence we have from the data that those proteins are part of a complex. So this is a small subunit of the mitochondrial ribosome and a large subunit of the mitochondrial ribosome coming together to make the mitochondrial ribosome, for example. And this is a subunit of the intraflagellar trafficking A complex that we solved a couple of years ago, that part of the ciliary machinery, or various other ones. So, the Hops Corvette complex, etc. So I've decorated a few structures out here to remind you there's this correspondence between these circles and real biochemical entities with structures and functions. The blue proteins are ones that we know the complexes for. And what we were really excited about is all the yellow ones are actually new uncharacterized protein complexes or new protein complexes that are falling out of our data. So we're seeing a lot in central metabolism, for example, of big assemblies of metabolic machinery, as well as in a lot in all of the sort of vesicular trafficking machinery that was so rich in LICA. And so we're spending a lot of time digging into specifics of these to follow up on them. But I'm going to show you something else that we do with this data to give you a sense of connecting it back to the kind of the human genetics. So we have this definition of the interacting proteins and the complexes, and we can essentially now map information about human genetics onto the interactome and then start finding new causal genes for genetic diseases using guilt by association. And we do this initially quantitatively. We test all those human gene sets linked to diseases to see how well they're recapitulated by the interactions. And for the ones that are well predicted, then we assume that the interactome is capturing the elements of that disease well, and we look at new candidate genes based on the interaction. So here's an example. These two half-black circles are known causal genes for osteopetrosis. So probably, I would guess a lot of you know osteoporosis. That's when your bone density drops too low, your bones get fragile, they break easily. So osteopetrosis is just at the other end of the spectrum. This is your bone density gets too high and your bones get brittle and they break easily. So for bone density, you really want the sweet spot. You don't want it too high and you don't want it too low. But osteopetrosis genes are interesting because the thinking is that if you had a therapeutic targeting an osteopetrosis gene, such that targeting that gene increased bone density, that might be a treatment for osteoporosis. And so there's sort of interest in being able to discover more of these genes. And so we prioritized new candidates. That's these half blue circles here. And our top candidate was this ATP6V1A. And we got ridiculously lucky here because the International Mouse Knockout Consortium had actually made a knockout mouse, and specifically the COMP2 group at Baylor College of Medicine knocked out that gene and then looked using X-ray densitometry at the bone density of those mice. And sure enough, they have strongly elevated bone density in the knockout mice versus the control mice. So bona fide osteopetrosis gene that we're able to identify on the basis of the protein-protein interactions in the map. And the reminder being that this is, in theory, this is the LICA interactome. But of course, these are conserved interactions that are shared across many modern eukaryotes. And so LICA wouldn't have had osteopetrosis, but these are all getting at lysosome acidification and aspects of that, which is important for bone mineralization. And so LICA certainly had vacuoles and the associated acidification machinery, and that was inherited. And in the mammalian lineage, this turns out to be linked to bone density in the contemporary mammals. So by interrogating with known osteopetrosis genes, we're kind of setting what we want back in our query, and it's giving us things that should have that property on the basis of these conserved interactions. I'll give you a second example of this. In the course of working on the map, we were contacted by clinical investigators at Boston Children's, Shirley Schrill and Friedhelm Hildebrandt, and they had a patient, neonatal patient, present with end-stage renal failure and other sort of associated features. And they sequenced the child and the family, and they could identify a variant in a very poorly studied gene called EFHAN C2 protein. It was a single amino acid change. And there was really nothing known about this gene, but for a piece of evidence that came from green algae, that it was involved in the cilia of green algae. And so they reached out to my colleague, John Wallingford, who, because he works on ciliopathies and knows a lot about cilia and frequently tests them alleles in animal models, they reached out to see if he could shed some light. And we were in the middle of working on the LICA map, and so we looked up EFHAN C2 in our LICA map and found that we did have evidence for it. It is one of these LICA orthogroups that goes all the way back a couple billion years. Its interaction partners do too, and the interactions are entirely ciliary in nature. So maybe it does other things, but the only evidence we could find for it was that it had a strong conserved function in the cilia in some fashion, and with evidence across different species. And when we looked at the multiple sequence alignment of EFHAN C2, the mutation in the child changed a completely invariant amino acid residue. This is across the organisms in our LICA tree here, completely invariant residue in an ancient protein module linked to cilia. And so it seemed likely that this was really kind of going to be the key point of failure for this protein. And so Chanjay and John's lab GFP tagged the protein and stuck it into frogs. And there's a nice assay on the skin of frogs as a multiciliated epithelium. Here you can see these cells outlined in a membrane marker, and these tufts are the cilia. And if you stick in EFHAN C2 with the GFP tag, it goes straight into the cilia, so very nicely localized. But if you add the child's mutation, it completely blocks the expression within the cilia. And so we're pretty certain that this is affecting this ciliary role, turns out to be critical for kidney development. And when we look in the interactome, the two interaction partners here with green half circles are actually already linked in the literature to renal disease problems, and one of those to end-stage renal disease. So we have interacting partners all linked to renal function in the ciliary module. We don't know how this gives rise in a cellular sense to the problems, specific problems with the kidney, but at least we have a strong starting point to move forward with. So this is sort of how we're using this information in a genetic sense. Now, moving forward in a structural sense, what we've been trying to do is to get richer information about these systems that's more fine-grained. And we've really dived down and focused on the cilia specifically. Just for perspective, this is about 5% of the human genome. About 1,000 genes contribute to making these cilia. So these long microtubule projections out of your cells, nearly all your cells have them covered in a plasma membrane with active motors trafficking things in and out, very complex dynamics in the cell to manufacture them, and many linked to ciliary diseases. And what we did from our biochemical separations is we realized that we had great data from Tetrahymena, this little microbe that my daughter calls a hairy avocado. So we can essentially shave the hairs off. We add a particular anesthetic, and they'll, from a liter of Tetrahymena, they'll drop all the cilia. And then Katie and Ophelia and helpers in the lab could then add chemical cross-linkers to the cilia that would cross the membrane, and any place there were two lysines within 30 angstroms of each other, we had a hope of being able to covalently couple them and then read out the couplings, the cross-links by mass spectrometry. And in doing this, we ended up mapping about 4,800 specific, unique lysine-lysine cross-links across all of the ciliary proteins. And this gives us very strong evidence, not only for the interactions, so we can confirm our big map, but it also now gives us detailed information for structural modeling. And where we've been getting a lot of power in this is actually in combining it with cryo-electron microscopy. So this would be a little piece of the interactome at the level of proteins, but what we'd like to do then is move to molecular models. And so we actually teamed up with Hui Bui, and Katie in particular worked with Hui's lab to take the cross-linking data, and Hui's lab had collected these just beautiful cryo-EM data sets for different aspects of the cilia. This particular one is in the cilium with these long microtubule kind of bundles, doublet microtubules around the periphery of the cilium. There's regulatory complexes that sit between those. This is one of those regulatory complexes called the nexadenine regulatory complex. You can see these long coiled coil proteins within it. These can actually be quite hard to assign in the cryo because there's many different coil proteins participating. And we found that the cross-linking data was especially helpful for being able to combine computational models, so think AlphaFold models, with the cross-linking evidence, that's these little black bars here, to make sure that we have the identification and the registry correct on the model building. And so using all of this information together, Hui's lab was able to model all 15 subunits in this nexadenine regulatory complex and get the structure. And then now has applied this in other contexts as well. And the really striking one is inside the doublet microtubules themselves, there's proteins lining the interior that are part of their functionality. This is again from tetrahymena, but many of these are conserved in humans. They were able to get a nice cryo-EM structure of these microtubules and see all this extra density inside. And then the challenge is there's a lot of different proteins that are participating in this process, which is which. And you can tell it's hard from all of these long coil ones, or single helix and so on are difficult to identify. So the general workflow is something like from the cryo, if there's a protein of known structure, homology model that. If it's not a known structure, collab fold to predict a structure. And then from the density, build in backbone traces, and then there's a neural network algorithm called Find My Sequence that'll attempt to match the deviations in the main chain density with likely amino acid sequence identification. And then all of this disambiguating and validating using the crosslinks. And on the basis of that, they could build a just beautiful model of 42 separate microtubule interproteins. I like this as a case of the challenges that we're going to face moving towards full structural information across the entire human proteome. As you start getting into these large, very large multi-protein assemblies, these are going to be wickedly difficult by pure computation. So I'm a big fan of the experimental constraints in combination with computational methods as a path forward for these really complex cases. This also convinced us that we should be taking a lot of our biochemical fractions and just throwing them onto the electron microscope and visualizing them. And so for interesting fractions, like ones where we're looking at larger things, where we think there might be distinct shapes that we could actually try to identify, Katie and Gabe started tossing some of the fractions mid-COVID. You can still see they're wearing masks there onto the EM. And from that, we get these sort of very complex micrographs of the contents of the fraction. So we know the identities by mass spec. We know the organism we purified it from and what the separation was. And you can see all of these discrete complexes, but it's still a complex mixture. There might be 100 different complexes in here. But we can still computationally cut out what look like discrete particles from these images and then cluster and average them to try to get a sense for what we're reconstructing. And in doing so, this is cilia from tetrahymena again, we realized we kept running into the same structure all the time, this kind of football-shaped thing. So I'm going to wrap up by telling you what this is, and then we'll leave lots of times for Q&A at the end here. But we ended up going and reconstructing this structure. And in doing so, we discovered an entirely new protein complex that we didn't know existed before. And we've called this the cage complex. So it's a hollow megadalton protein assembly that we purified out of the tetrahymena cilia. We initially solved a 7 angstrom structure from tetrahymena. We figured out what the protein was because we had mass spec identities and so on. We could model it with AlphaFold, and this is a great case of a out of training set prediction where AlphaFold actually did quite a decent job. A number of the folds in this are entirely novel, and AlphaFold was able to do a good job on those folds. It tends to not do such a great job positioning the domains relative to each other, but for the individual folds themselves, it was actually quite respectable. And we could test all of this with our cross-linking mass spec again, and then go hunting for more instances of this. And actually, across the full tree of life, we find this has a really peculiar phylogenetic distribution. So we called it cage because of the shape, but also it's a conserved assembly in gram-negative bacteria and eukaryotic microbes. We haven't found it yet in any complex multicellular organism, so it looks like it was most likely a LICA, but it was lost in animals, so we don't have it. But the sort of the distribution of a few thousand homologs is it's in a giant clade of bacteria called the PVC clade. So these would be the plankton mycetoda would be the bacteria in the ocean that make gaseous nitrogen with a membrane-bound organelle called the amoxosome. Very cool bacteria. And then on the eukaryotics side, we see it in green algae and excavates and choanoflagellates and ciliates and fungi and so on, but not, for example, in animals. But this did give us new sources, and slime molds are fascinating and interesting creatures. So we grew up a bunch of slime molds, and we solved the structure out of the purified endogenous one from slime molds. So this is now closer to a 3 angstrom structure of the cage. And for scale, this is the width of the large subunit of the ribosome. So there's a little under 10,000 amino acids here. It's actually 4 copies of a roughly 2,400 amino acid protein make up the whole thing. And we have absolutely no idea what it's doing. So here it is relative to a few other big assemblies. Here's cage, and for scale, here's a few other ones. This is a synthetic mini carboxysome. The real one would be big, but this is here to see the geometry. It's about the same size as the GROE-L-GROE-S complex, and we're guessing that probably it might do something similar. So when you see cages of this size and shape, you tend to think transport or chaperone. So maybe it's a new class of protein or RNA chaperone or transport machinery. But anyway, new fold, unknown function, and a reminder of just how many mysteries there are out in biology. This is yet another component of LICA, in this case lost on the human lineage, but another piece of this ancestral machinery there. Okay, so I was given strict instructions that I have to wrap up and take questions. So I'm going to end here. I'm going to once again thank all the people that contributed to this and who I've tried to call out as we go along the way, and I'll stop and take questions.